I'm Curt Sigmund. I am the immediate past chair of the Council on Hypertension and the chair of the Excellence Award Committee of the Council on Hypertension. I'm also from the Medical College of Wisconsin, and I'm here to introduce Dr. Bina Joe, the 2024 recipient of the Council on Hypertension Excellence Award in Hypertension Research. So the Excellence Award is the highest honor bestowed upon by the Council on Hypertension on a scientist who has made extraordinary contributions to our understanding of blood pressure regulations and the mechanisms of hypertension. Dr. Bina Joe is the Distinguished University Professor and Chairperson of the Department of Physiology and Pharmacology and the founding director of the Center for Hypertension at the University of Toledo. And she's receiving this award for seminal contributions toward understanding the genetic mechanisms of hypertension, the discovery of gut microbiota as a key contributor to blood pressure regulation, the ketone body beta hydroxybutyrate as an antihypertensive metabolite, a link between energy metabolism and hypertension, and engineering microbiota as a therapy for hypertension. So Dr. Bina Joe, congratulations on this honor. And maybe you could tell us a little bit about what you're going to talk about in your presentation tomorrow.
Happy to do so, Curt. First of all, it's wonderful to be talking to you, who's a previous recipient of this award, and on the 100th birthday of the American Heart Association. So it's a very special year to receive this award, and I'm honored. Tomorrow I'm going to be talking about the microbiota. The focus is going to be on the microbiota per se, among all the things you mentioned, because of the time that we are provided to give this lecture. And there'll be three topics that I'm going to hit. One is on salt sensitivity, and we're going to reveal the fact that we have generated a germ-free SRAT model, which is a fantastic tool for the field of microbiota research. And the second, I'm going to talk about our work on glyphosate, which surprisingly is pro-hypertensive. It may raise some eyebrows, but it's probably a mistake that we have made in the previous century, not knowing that we are holobionts. The premise was to develop something that wouldn't hurt us, but would kill plants, weeds. But it happens so that the biochemistry is such that these pathways are present in microbes as well. So we have changed our internal climate of this factory of microbes, perhaps by using these products on our food. And the third I'm going to talk about is engineering of microbiota as a proof of concept for potential use of microbiota as medicine.
So Bina, you've made extraordinary contributions not only to the genetics of hypertension, but also in this newer area of yours in the microbiota. And I'm wondering what inspired you to make the shift in your research interest.
It's a great question. Things come to you. It wasn't by choice that I went after the science. Working on the genetics, we now know that we haven't hit that magic bullet where genetics can be used as therapy. You know, there's minor alleles of thousands of them. So at that point, I was discussing with a colleague from the IBD field, Vijay, whom I've recruited into Toledo, about salt being antibacterial, and his work was on microbiota. So we combined those two thoughts and decided to do an experiment to see if a salt-sensitive animal would indeed be salt-sensitive because of its microbiota. And that's what triggered this in 2014, which is the dawn lecture when I announced that we did this experiment, and you were the one to say, hey, it's the bugs that are causing blood pressure. Yeah.
I do remember that conversation.
Yes.
So I'm wondering what kind of challenges or surprises, you've had along the way.
The biggest challenge is that you're alone when you start a new field. And to have the resources developed around it, we had to start everything from scratch, in addition to understanding the names of these bacteria and trying to understand how to arrive at 16S sequencing. So we bought all the equipment and had to get going on that front. And then it was fantastic to see that other model organism researchers picked up this thought and went after it. So there were publications coming. And the biggest hurdle was to convince our peer reviewers that this is great science. It still is a challenge when you start a new field, and we're not going to give up because this is big. We are not one organism. We are a whole organism with a factory of microbes that do things for us. And so viewing that as an ecosystem is a new thought for 10 or 20 years of science, basically, and only 10 years old in hypertension field. So the hurdles are still there. Having to pull talent into this field is a hurdle. And so I have developed a department with microbiota as a theme. And so several researchers picked it up, and we are on the get-go, from the get-go, on this mission to have more investigators join this thought. I will also say microbiome as medicine is perhaps the same ideology we had when we had the thought of genetics as medicine. It may or may not reach that status, but understanding the physiology of what microbes do, microbiota does to our bodies, is critical to know where we would go with this.
Thank you. I knew somewhere along the way funding would come into some of the challenges, because we all face the same challenges in that line. So you mentioned that you're building a department and you had to recruit a team of investigators. So maybe you could talk a little bit about how you were inspired by your mentors and how you are inspiring the next generation of researchers to, what kind of advice do you give to the junior folks that are in your department or in other laboratories?
Great question, Curt. Times were different when we were growing up. Maybe when you were growing up, it was even more different than when I was growing up in science.
Oh, we're not that different.
Well, I like that then. Point is, we didn't have training in grant writing. We didn't have training at networking. We just went with the science and understood that there were giants in the field that you could go and ask and learn from them. One of them was John Rapp. I owe my entire hypertension career to John Rapp because I used to previously work with arthritis at the NIH on the genetics of arthritis. So the switch was with John Rapp. He was also a previous awardee, a Novartis awardee. And in 2001, I joined his lab with the intention of actually going hook, line, and sinker deep into genetics to find genes. And even after his retirement, I continued that aspect of it with a lot of energy at that point. Now coming to nowadays, what's happening is there is a lack of talent that really wants to do science. And the ones that come in are burnt with the idea of, do I go to industry or academia? And the burnt idea is academia is hard. And that's the truth. But the point is, the ones who want to do it actually do it with a lot of interest. And there are youngsters like that. And when I get them under my fold, the way to train them is to give them independence and attach responsibility to it. They love independence, and when it's given to them, they become responsible. And that's the aspect of training that I hope people will appreciate and remember as a part that would be great, right? Research has to be done independently. And if they can do it independent of the PI at an early phase in their career, that's wonderful, isn't it? We have done that through even developing programs such as postdoc to faculty positions. Several of them have moved on to tenure track positions, and several at this meeting. And it's so much joy to see them blossom very early with such pathways, and they run with it. So I think they're smart. At the same time, we got to, make them understand that this is good science for some of them, for them to stay on in the hypertension field. That can be hard, but we'll never give up, right, Curt?
Absolutely. And I know from your former trainees how passionate you are as a mentor and an advisor and a coach and a friend to them as they develop their careers, and even after they leave your nest and move on to something something else. So my last question for you, you already mentioned during your early statement that this is the centennial
Yes.
celebration of the American Heart Association. So maybe you could tell us a little bit about how important the American Heart Association has been in the development of your own career.
The American Heart Association has been for 100 years complimenting all investigators that work in hypertension. That's a truth because this is our primary meeting, the Council for Hypertension, that we come to. And I've been coming to these meetings for decades, and I've seen several of you receive this Excellence Award and aspired to go there with my science. So it was motivational to come to this meeting first. Secondly, the grants that come in, the pre-doc grants especially, are very, and postdoc ones, are valuable. They are the stepping stones to many who end up as great professors. So that's a big part of it. And socially, to meet people at this meeting is a big thing. During COVID, that's one thing I missed a lot. It is the ideas that come through these meetings do convert into projects and do converts into potential therapies. It's all hand waving for some who are basic science researchers like me, but integrating with clinicians and brushing shoulders with them at this meeting has been helpful.
Excellent. So, Bina, my wholehearted congratulations once again, and we are looking forward to your lecture tomorrow.
Thank you, Curt.
Okay.